So without further ado, let's get this party started. Today I'm going to start off by giving you a little background into the element nitrogen. Then I'll talk about the basic components of symbiotic nitrogen fixation and the strategies used to accomplish this process. I'll follow with a brief description of invasive plant characteristics and ecological consequences of nitrogen fixing invaders. I'll get you up to speed on the current state of knowledge regarding these topics, and then I'll transition into my research, interest and focus. And next will come the details of my experiment, followed by a discussion about the implications of my results. We'll finish off our time together with any questions you may have or suggestions for things to consider that I may have overlooked. So to begin with the overstory, I'm sorry, I just need to make a couple little adjustments here on my screen.
Join the meeting.
Okay. Nitrogen is everywhere. Nitrogen is one of the most abundant elements, a large portion of which is in the atmosphere as N2, or nitrogen gas. Also in various compounds in the ground, in the oceans, in plants, and in animals. It is literally everywhere. Nitrogen is necessary for plants to grow. It's a component of chlorophyll, amino acids, which are the building blocks of proteins, ATP, which is the energy currency of cells, and nucleic acids. Although nitrogen is abundant, plants can only utilize it in reduced forms, so it is often the limiting factor for plant growth in primary production in terrestrial environments. Biologically useful forms can become available to plants through applications of industrial fertilizers or manure to soil, during organic matter decomposition, when lightning or volcanic eruptions convert nitrogen gas into compounds, and through biological nitrogen fixation. Symbiotic nitrogen fixation can occur between certain types of soil bacteria and compatible plant species, and is one of the most efficient systems for reducing nitrogen gas from the atmosphere, or fixing nitrogen. Bacteria infect the roots of plants, and together they form nodules. You can see circled here in the photo. Within the nodules, the bacteria use the enzyme nitrogenase to catalyze the reduction reaction that converts atmospheric nitrogen to ammonia to be delivered to the host plant. The host plant provides carbon-based metabolic compounds, generally sugars, and oxygen to the bacteria to power the process. Although this is a costly process for the plant, the trade-off is a competitive advantage, particularly under conditions of low soil nitrogen availability. Nitrogen-fixing plant species use either an obligate or facultative strategy, resulting in different amounts and rates of nitrogen fixation. Obligate fixation occurs at an unregulated constant rate per biomass unit, regardless of how much nitrogen is available in the soil, devoting a lot of energy and resources to fixing atmospheric nitrogen. Facultative fixers regulate fixation with changes in the amount of available soil nitrogen to meet the plant's needs, and are able to allocate conserved energy and resources to growth if there is adequate soil nitrogen available. For simplicity, we can think of the obligate fixers as unregulators and the facultative fixers as regulators. Depending on environmental conditions, both of these strategies have trade-offs and competitive advantages. Let me show you how these work. Let's imagine that these two leaves are nitrogen-fixing trees in the forest. One tree is an unregulator, and the other is a regulator. All plants demand a certain amount of nitrogen to carry out metabolic and other biological processes. For simplicity, let's say these two trees require the same amount of nitrogen, represented here with the purple line. The light blue shaded areas down here at the bottom represent the biologically available nitrogen in the soil, and the bright green shaded areas here represent the amount of nitrogen fixed by these specific trees. This is an example of a low nitrogen environment. The unregulator fixes a certain amount of nitrogen constantly, no more, no less. So in this N-poor environment, the obligate fixer is limited. The regulator, however, fixes as much as it needs to meet the needs of the tree. So in this nitrogen-poor environment, neither tree can grow very fast because one is limited and the other, or the one is limited in investing, and the other is investing a lot of resources into fixation. Here we see non-limiting levels of available nitrogen in the soil here, and the different responses of the trees. Nitrogen demands of the trees remain the same, but with more nitrogen available in the soil and a constant fixation rate, the underregulator now has more nitrogen than it knows what to do with. So, although some growing is occurring, no N loss via tissue turnover is also occurring. On the other hand, the regulator capitalizes on the available soil nitrogen, conserving resources that can then be allocated to growth. In an N-saturated environment with such high levels of nitrogen available in the soil, the unregulator not only gets plenty of nitrogen from the soil, but continues to fix at a constant rate, leading to more tissue turnover and nitrogen loss. The regulator is stoked because the soil is providing enough nitrogen to meet all its demands, and it can allocate a majority of resources to rapid growth and reproduction. Invasive plant species are non-native species that have been introduced, perhaps for horticulture or agriculture, for example. They naturalize and increase rapidly outside their home range. Invasives tend to have greater growth and performance rates compared to natives and non-invasive alien species. Phenotypic plasticity is a characteristic of invasive species and generally implies that a species can sustain fitness in unfavorable conditions and or increase its fitness when conditions are more favorable, meaning that the species can actually change its morphology in response to changing environments in order to be better suited for the environment. These trade-offs allow invasive plants to outcompete natives and non-invasive species, particularly in areas with high resource availability, like disturbed environments and areas in frequent flux. Invading fixers have the potential to disrupt entire ecosystems during development and processes, and many invasive plant species are nitrogen fixers. Invasives can assist other non-native plant species to successfully invade, often with devastating consequences for native forests and ecosystems. By contributing more biologically available nitrogen to soils, altering forest structure, creating changes in the amount and quality of litter, and contributing to elevated enzyme activities in the soils, positive feedbacks in biogeochemical processes can alter outcomes for developing and existing ecosystems. We have witnessed these consequences here in Hawaii with the invasive nitrogen-fixing trees, Morellifia and Facultative moluccana. Hawaii has adopted a risk assessment system based on the Australian and New Zealand risk assessment system. The assessment renders a numeric risk score that considers factors such as a species' history of invasiveness outside its native range, favorable environmental conditions in the new location, life history events such as persistence, reproduction, and dispersal, and the ability to fix nitrogen to predict. The assessment is used to predict whether a species will be a pest if it is introduced to the islands. Plant scores range from -12 to 28 and generally fit the scale shown here. If scores are below 1, they are considered low risk for invasion. Above 6 are considered high risk, and in between require further evaluation. Lots of research has been devoted to nitrogen fixation and even more to invasive species. A meta-analysis investigating the mechanism for invasiveness looked at over 100 studies that compare natives to invasives and invasives to non-invasives in the invasive range. Concluded that, in general, invasives have higher performance trait values than non-invasive alien plant species, but they do not explore nitrogen fixation at all. Another meta-analysis investigating fluctuations in the nitrogen pool with invasion did include nitrogen-fixing invasives in their analysis, concluding that nitrogen fixers do have a role, for example, accelerating nitrogen fluxes and contributing more nitrogen to pools, but they did not explore differences in nitrogen fixation itself, for example, strategies and or rates. They only differentiate whether species are non-nitrogen fixers or not. More recently, a review of alien plant research with emphasis on patterns, drivers, biotic interactions, and evolution suggests that some alien plants are likely to become invasive if they participate in symbiotic nitrogen fixation, especially if nitrogen fixation is not already present in the native species. Again, a yes or no question of whether fixation occurs in the plant. No studies have investigated fixation strategies of invasive plant species compared to non-invasive alien plant species, or the relationship between fixation strategy and invasiveness. It is possible that the strategy used for nitrogen fixation, rather than the ability to fix, explains the differential success of non-native nitrogen-fixing plant species. If so, exploring fixation strategies among invasive and non-invasive alien plant species is an important step to understanding invasion and forest dynamics. And this is the focus of my research experiment. So we move on to the understory. My study followed closely to the methods and modeling presented in Menges et al. 2015 study of the diversity of nitrogen fixation strategies in Mediterranean legumes. In order to determine the strategy of nitrogen fixation in plant species, several aspects of the plant need to be assessed. Growth rates, biomass allocation, well, you can see them listed here. Experimental greenhouse studies are conducted in order to keep environmental factors such as temperature, light, and precipitation constant. With these factors constant, we can isolate nitrogen-fixing plants' responses to nitrogen availability by providing nitrogen-limited to nitrogen-saturated conditions while keeping other nutrients constant and in adequate supply. The isotopic signature of biologically available soil nitrogen is different than that of the atmospheric nitrogen. Whether nitrogen is derived from the soil or fixed from the atmosphere can be determined by using an isotopic tracer administered through fertilizer treatments, followed by isotope ratio mass spectrometry. These data allow the strategy used to be distinguishable. Obligate strategy users cannot regulate fixation. Facultative strategy users regulate fixation so that nitrogen supply meets the nitrogen demand of the plant. To explore strategy and trait differences of invasives and non-invasive alien plants, I used weed risk assessment scores as an index for invasiveness and chose 4 high-risk and 4 low-risk species across a range of numeric scores, as well as a non-fixing high-risk species for reference, all within the family Fabaceae for my experiment, which leads me into my research questions. Do growth rates, allocation traits, and nitrogen fixation traits vary among non-native nitrogen-fixing tree species that have a range of assessment scores? Is there an association between the weed risk assessment scores and fixation traits? What fixation strategies are these trees using? And do species that exhibit greater trait plasticity have more or less regulation of nitrogen fixation? In general, I expected that high-risk species would have more variability in response to nitrogen addition than low-risk species. For example, I expected that the difference between growth rates at low nitrogen and high nitrogen addition would be greater for the high-risk species than the low-risk species. I also hypothesized that the high-risk species would have decreased fixation and nodule density with nitrogen addition, evidence that would be consistent with a facultative regulator strategy, and that low-risk species would have little or no change in fixation and nodule density with nitrogen addition, evidence consistent with an obligate unregulator strategy. Finally, because trait plasticity is a characteristic of invasiveness, I expected high-risk species would have greater trait plasticity than low-risk species, and hypothesized that trait plasticity would correlate with fixation regulation. In other words, that the high-risk species would be facultative fixers with the greatest trait plasticity in response to nitrogen addition, and low-risk species would be obligate fixers with the least trait plasticity. Moving on to my methods. In a shade house experiment, 8 nitrogen-fixing tree species within the family Fabaceae were grown, all of which have been evaluated using the Hawaiʻi weed risk assessment system. 1 non-fixing reference species from Fabaceae was grown under the same conditions for comparison. 4 high-risk and 4 low-risk species were selected for experimentation, and 1 high-risk non-fixer. Species within the evaluate category were not studied to avoid confusion. 3 levels of nitrogen addition were given for each species, beginning with 16 replicates per nitrogen treatment per species, only 12 for Erythrina variegata though, in a randomized block design. Study species were nitrogen-fixing tropical tree species chosen based on their weed risk assessment scores. Hereafter, study species will be referred to by their abbreviations. I just want to point out that the font color here, the green are the low-risk species and the red are the high-risk species. And also note the wide range of assessment scores. Seeds were collected on Hawaiʻi Island, except 2 low-risk species were purchased from overseas because I just couldn't find enough low-risk species seeds here. Seedlings were inoculated with bacterial symbionts at approximately 2 weeks after germination. In a shade house, saplings grew for approximately 4 months after the first fertilization. Fertilization consisted of a standard nitrogen-free fertilizer to keep other nutrients constant, and 3 levels of isotopic nitrogen fertilizer to mimic an N-limited environment, an N non-limited environment, and an N saturated environment. Final growth measurements were taken for each plant. All components of the plants were harvested at the end of the growth period. Leaf area was measured for fresh leaves. Biomass measurements were taken separately for each plant part. And because the plants were grown for different periods of time, ranging between 115 and 132 days, all biomass data are represented as growth rates per year to account for variation in growth time. Samples were then pooled per individual for isotope analysis with 10 replicates per treatment per species. Calculated variables included allocation ratios and percent NDFA, and also trait plasticity. Plant biomass is an indicator of the effects of nitrogen limitation on plant productivity. Root nodule mass is a measure of structural allocation to nitrogen fixation, and percent NDFA indicates how much nitrogen has been fixed over the lifetime of the plant. It means nitrogen derived from fixation. To investigate differences in species means for growth rates, allocation traits, and nitrogen fixation traits, linear models and ANOVAs were performed, linear models or two-way ANOVAs were performed on 2 factors, species and nitrogen treatments, followed by Tukey's on a significant difference post hoc comparison tests. Log transformations were made when needed to achieve normality. A principal component analysis was performed on all response variable means to explore species grouping, and regression analysis was performed to investigate the relationship between trait plasticity and nitrogen fixation. Moving forward onto the results. My first question asked if growth rates, represented by biomass up here on top, allocation traits, represented by root to shoot ratio here in the middle, and nitrogen fixation traits, represented by percent NDFA on the bottom, vary among species with a range of assessment scores. Species are on the x-axis here and are in order of their assessment scores, lowest to highest. Low-risk species are in green, high-risk species are in red. Response variables are on the y-axis and are representative of other measurements taken. Means are represented by solid red triangles here in the middle. Whiskers show the highest and lowest point data values, excluding outliers. And letters up here indicate significant differences among species, with p less than 0.05 and alpha at 0.05. So the answer to question 1 is yes. Yes, growth rates, allocation traits, and nitrogen fixation traits do vary among species. However, there does not appear to be an association between assessment scores and fixation. So the answer to question 2 is no. As you would predict with nitrogen fixers, biomass increased with nitrogen addition, shoot allocation decreased, and percent NDFA decreased. All measured variables showed a significant difference in means across the nitrogen addition, except for specific leaf area. Moving on to the interaction of the main effects. The effects of nitrogen on growth rates and nitrogen fixation traits, nodule density, and percent NDFA. Again, species are on the x-axis in the same order as they were before, with the low-risk species in green and the high-risk species in red. Response variables are here on the y-axis. Growth rates on the top, again, nodule density in the middle, and the amount of nitrogen fixed on the bottom. Growth rates varied across species with nitrogen addition. Most species had a reduction in nodule density with nitrogen addition, and all species had a reduction in percent NDFA with nitrogen addition. These interactions help determine which fixation strategy each species is using. To determine fixation strategies and answer my third question, I plotted nitrogen fixation traits, seen on the y-axis, against nitrogen addition, the treatments of the experiment. On the y-axis, again, we have biomass. Well, we have biomass nitrogen at top, nodule density in the middle, and percent NDFA at the bottom. Note the difference in scale on the y-axis. The blue triangles here represent species means for nitrogen fixing species, and the orange triangles represent the non-fixer SESI. Error bars are standard error. In comparison with the non-fixer, 3 patterns emerged. SASA was determined to be the only obligate strategy user. SASA's biomass decreased with nitrogen addition. However, nodule density remained constant, and while percent NDFA slightly decreased, it was a very minimal decrease. The facultative strategy was determined for each of the other species. With an increase in biomass nitrogen across nitrogen treatments, the other 7 species regulated nodule growth and N fixation to some extent, shown by reductions in nodule density and percent NDFA. 3 of the species regulated fixation so much that percent NDFA was actually calculated to be below 0 and appeared, so these species appeared to be over-regulating. All 8 study species fell into one of these strategies. Another way of looking at this data, using a principal component analysis, we're able to see how species are grouping in trait space. Down here, PC1 on the x-axis, going from low to high, was associated with growth and allocation traits and explained 49% of variation among species groups. PC2 here, going from low to high, was associated with nitrogen fixation traits and explained an additional 32% of the variation. Recall that Sasa, a low-risk species at the top in turquoise, was the only species using the obligate strategy and appears to be all by itself with high fixation and only mediocre growth and allocation. However, FAMO, PIDU, and SEGR, these 2 are high risk and this is a low risk, were the 3 species that appeared to be over-regulating and are grouped here with lowest fixation. Aaron, which is a low risk, had super high growth rates but was not able to regulate fixation as well as most of the others. So this figure shows that traits relate to fixation strategies and how strategies can be mapped out based on traits. To answer my 4th question, I ran a regression analysis for trait plasticity in response to nitrogen addition and the nitrogen fixation traits, nodule density up top, and percent NDFA down here. Plasticity is on the x-axis and is greatest at the left end of the plots. Fixation traits are on the y-axis, nodule density and percent NDFA. And sure enough, I found a positive relationship between trait plasticity and fixation regulation. Sasa, circled in turquoise, again, the low risk, has the least trait plasticity but the greatest nodule density and percent NDFA. SEGR, FAMO, and PIDU, circled in orange, have high trait plasticity and low nodule density and percent NDFA, or high regulation. In other words, the species that have the most plasticity regulated fixation the most. So a real quick summary of so far what I've said. My study revealed that the 8 nitrogen fixing tree species varied in growth, allocation, and nitrogen fixing traits and could be grouped into 3 distinct fixation strategies. Strategies appear to be influenced by the trait plasticity of each species, but do not appear related to the weed risk assessment scores. So moving on to the discussion. Sasa is a low-risk species that demonstrated the obligate strategy, meaning that it did not adjust fixation even when more soil N was available. It has been introduced and naturalized throughout the tropics and is not a high threat for invasion. Obligate fixers tend to do poorly in tropical ecosystems, which are typically not nitrogen limited, because the investment of resources into long-lasting nodules that fix nitrogen at a constant rate is too costly to compete with coexisting non-fixers. In nitrogen-rich environments, obligate fixers overfix, leading to N losses via tissue turnover, whereas in nitrogen-poor environments, they underfix, leading to nitrogen limitation. Obligate fixers can do well in tropical ecosystems during early succession if nitrogen availability is low, but after enriching the soil via fixation and litter fall that they do themselves, they will likely be outcompeted by non-fixers. So obligate fixers are most successful in environments that have large nitrogen losses frequently. Within the facultative strategy, there can be 3 strategy distinctions. Overregulation, so that with increased soil nitrogen availability, fixation is downregulated so much that less nitrogen is fixed than is needed to meet the plant's demand, making it N-limited. Underregulation, where increased regulation occurs with increased soil nitrogen availability, but regulated to the point that fixed nitrogen is greater than the needs of the plant. And incomplete downregulation, in which there is some downregulation, but fixation rate will stay constant in all soil conditions. Overregulation was the only variation of facultative fixation found in my studies, so that's what I'll focus on here. Overregulation was clearly observed in Symbiotic grandiflora, Facultaria moluccana, and Physiological dulce, by their negative values for percent NDFA with high nitrogen treatment. Symbiotic is native to South and Southeast Asia. Facultaria is native to the Moluccas in Indonesia, and Pithecolobium is native to Central America. All of these species have been introduced throughout the tropics and are considered highly invasive on tropical islands outside of their native ranges. However, I would like to point out that Symbiotic has been evaluated as low risk here in Hawaii. The facultative fixers had minimal and inconsistent variation in response to nitrogen addition for growth and allocation, but all reduced fixation with nitrogen addition. Erythrina variegata is not considered a threat outside its native range. Pterocarpus indicus is not known to threaten intact native plant communities, but is invasive in disturbed environments. Albizia lebec is highly invasive throughout the tropics, and Acacia confusa has not been studied enough to conclude how invasive it is, but it is considered invasive here in Hawaii. The most successful facultative fixer regulates fixation just enough to be co-limited by other nutrients and have the highest growth rates, relative growth rates. This sounds ideal, so why aren't all facultative fixers invasive? Having the internal mechanisms to adjust fixation, for example, making or destroying nodules or activating or deactivating symbionts, to keep up with the changes in the local environment, may come at a cost that slows growth rates and or hastens tissue turnover. In addition, time lags may play a role in hindering facultative fixers because nutrient conditions in the environment change very quickly, and fixation regulation may take more time to kick in. And when fixation is not regulated to perfection, even a small amount of time over or under regulating can result in nitrogen loss, creating elevated nitrogen pulses that can lead to long-lasting nitrogen richness in the environment. Both the costs and the time lag scenarios can decrease the competitive advantage of the facultative fixer. They are most successful if the costs of being facultative are low and the environment is nitrogen poor. A couple of things to consider with my findings. First, the overregulators had negative values for the amount of nitrogen derived from fixation. However, NDFA does not drop below 0 as far as I know. Either there is fixation resulting in a positive value or there is not, resulting in 0. Overregulation implies that fixation is regulating so much that the plant becomes limited by nitrogen, but the 3 species that appeared to be overregulating had increased growth rates and shoot allocation. These behaviors lead me to believe that using a representative non-fixer, which I use SESI, for the soil end member of the percent NDFA calculations is probably not the best method. Second, underregulation may have been observed in P10 and Ale, because although they had no change in growth rates and shoot allocation, they did have significant decreases in nodule density and percent NDFA with nitrogen addition. So this implies that there was some regulation occurring, but perhaps not enough to maintain tissue and increase growth. More refined methods and calculations would help conclusively answer these questions and concerns. I found that there was a positive relationship between trait plasticity in response to nitrogen addition and the ability to regulate fixation. Greater trait plasticity generally implies that a species can sustain fitness in unfavorable conditions and or increase fitness when conditions are more favorable. Invaders tend to have greater plasticity and fitness traits in response to environments that correspond with the plastic traits. Being nitrogen fixers, we would expect the species in this experiment to be invasive if their nitrogen fixation traits were the most plastic and the environment was nitrogen poor. In other words, they would utilize a perfectly facultative strategy, allowing them to coexist with natives adapted to the nitrogen poor local environment and outcompete them through resource conservation and allocation. FAMO and PIDU both appear to fall into this specific situation. They were found to have high trait plasticity, utilize an efficient facultative fixation strategy, and have successfully invaded, meaning coexisted and outcompeted natives in nitrogen-poor environments in Hawaii. Although I did determine a diversity of fixation strategies in plants with a wide range of weed risk assessment scores, I did not find association between fixation traits or strategies and assessment scores. Several reasons may contribute to this finding. First, the Hawaii weed risk assessment system is not a continuous scale from lowest scores to highest scores and does not consider interactions between plant functional traits. Rather, it asks a series of yes or no questions about the plant's life history traits, native geographic and climate range, and history of invasiveness outside its native range, and generates an additive score. Answers to these questions are derived from scientific literature and experts in the field, and assessment scores using this system should ultimately be considered a prediction of whether an introduced plant is likely to become invasive in Hawaii, rather than as a scale of how invasive a species is likely, rather than a scale of how invasive a species is. Second, there is only one question on the weed risk assessment that asks whether or not a species can fix nitrogen. Nitrogen fixation itself does not determine whether or not a species will be invasive, but the strategy used for fixation may carry more weight than just a yes or no question. So a few concluding remarks. In tropical forests, different nitrogen-fixing tree species fix nitrogen at different developmental stages or age and at different rates, implementing mostly facultative strategies. Fixation strategies are inherent in and vary across individual species, and fixation occurs in response to growth and resource availability. This is the first study, to my knowledge, to seek to determine nitrogen fixation strategies for invasive and non-invasive alien plants, intending to answer the question of whether fixation strategy is related to invasiveness. I was only able to observe 8 species, and it's still unclear whether fixation strategies contribute to invasiveness, so more studies are needed to conclusively answer this question. And with that, I would like to thank my biggest supporters. First and foremost, Fern, thank you. Elizabeth, thank you for your patience. Joanna and Manon, you know what you did. And of course, Becky, thank you so much for all the hours and help and patience you've poured into me. John and Jen, also, thank you. Molly Murphy, thank you so much for all the help with the seed collection and data collection and planting and greenhouse moving and harvesting. And Chuck, thanks for your help with the weed risk assessment. This time I'd like to take any questions, if anyone has any, and open up the room to discussion.
Yes, I can help facilitate questions. But anyone have a question they want to be the first to ask?
I'm just going to shut my front door really quick because it started raining and it's loud. I can't hear.
Any questions? There's also a way I think you can raise your hand in the chat room as well.
Woo. Right. No questions?
Anybody?
Really?
I have one, but I want to see if others have anything they want to ask.
I just wanted to say, good job, Angie.
Oh, thank you. Thank you.
Okay, I have a question if others don't have questions. I'm wondering, do you think that you only had one obligate? It turned out you only had one obligate species and you didn't know that from the beginning, so you really had no way to know that. But do you think that means that obligate species are pretty safe bets and unlikely to become invasive? Or what do you think about that?
It seems like in the tropics, facultative fixers are, that's more their habitat, and the obligate fixers are more in temperate regions. And I think that has something to do with the bacteria that they're symbiotic with. But I think that I think that obligate fixer here would probably be pretty safe to not invade. I mean, their fixation depends so much on environmental conditions, and the conditions have to be just perfect for the obligate fixer to be successful and, you know, establish a community. That I don't think you should bring any new plants to Hawaii, but if you're going to bring a fixer, obligate's better than facultative for sure, in my opinion.
Other questions?
I have a question. Were all the most, the plants with the highest trait plasticity, were those only high-risk plants? Did those turn out all being in the high-risk category?
No, actually one of them was a low risk. Let me pull up that slide. So this is more plastic, this is less plastic. This species right here is actually a low risk species here in Hawaii, but it's considered invasive on other tropical islands. Mm-hmm.
Ange?
Yes?
So nice job.
Thank you.
So I was thinking about the native nitrogen fixers, and the only one that I know is koa, at least off the top of my head. Are there any others, and do you know what kind of nitrogen fixers they are?
There are others. I don't know what strategy they use. I'm not sure if that's common knowledge, although Jen may have an idea what some native strategies are. I imagine that they are probably facultative, but I don't know. I don't know. That's a good question.
I will just say that one of the things that's always fascinated me about the Hawaiian native legumes is just how incredibly different they are. So mamane, you know, an important nitrogen fixing tree in Hawaii, has the slowest growth rate of any plant that I've ever measured. Like, you know, you can have like 2-year-old, 3-year-old individuals that are just a couple inches tall. So, I mean, there's clearly something going on. And for a long time, I thought it might have something to do with fixation, because all the Mamani seedlings I've ever pulled up, I've never found any nodules on them. So I always thought nitrogen fixation could be really important there. But I guess I wanted to ask Ange a question about any sort of like phylogenetic analyses that you thought maybe about doing, or any sort of link with that. So Fabaceae is a pretty big family.
Mm-hmm.
So I wonder, you know, if you were to, I don't know if you've looked at this, but if you tried to map phylogeny onto some of these patterns, do you think that maybe like shared evolutionary history can explain some of these differences you're seeing across species?
Yes, I definitely thought about this, and this is something I'm hoping to look into over the next week or two, but I haven't had time to look into that yet. But it's definitely an interesting question, and I have thought about, you know, I'm looking forward to looking into it for sure. Do you have any recommendations to that end?
Yeah, we can play around with it. I'm kind of learning how to use these methods myself, although I'm sure there's probably someone on this call who's has more experience with that than I do. But yeah, I mean, it could explain, you know, some of the differences. I mean, right now we have this kind of arbitrary split with risk score, but kind of looking to see how phylogeny can explain some of these patterns could be interesting too.
Mm-hmm.
I have another question. Were the 3 levels of regulators, were those distinctions that were already known in the other literature or the other people you worked with, or is that a distinction that you kind of discovered through your testing?
No, I did not discover that. So actually, the paper that I used to model my study after, let me try to find that slide real quick. This one, you mean the overregulation, underregulation, and incomplete downregulation? Is that what you're talking about? Yeah. So the Mediterranean legumes paper that I mentioned in 2015, which Jen was a part of, they actually proposed these subsets, I guess, or I don't know, subcategories of facultative based on what they had found, and actually came up with models for how you can predict these different types of regulation within the facultative strategy. So I tried to mimic what they did, and then tried to you know, determine strategies among the species that I experimented with based on the models that they had provided in their study, in that study in particular.
I have a quick question. So there's no quick and dirty way to tell if something's obligate or facultative. You have to go through this isotopic tracer method to really distinguish it.
I mean, I think to get the nitty-gritty quantitative result, probably yes, but you know, it seems like the morphological plasticity is can probably point you in the direction of, you know, deciding whether it's facultative or obligate, I would say.
Okay. Other questions that people have? Anybody? Okay, last call. Any other questions?
Thank you, everybody, for coming. I really appreciate your support.
Ange. Yeah, we can all
So stoked right now.
we can all clap in our in our own place, even if she can't hear it all.
Thank you. Thank you. I love you all. Aloha.
Thank you so much for coming, and this will be recorded if someone wants to watch it at another time. And thank you for the comments as well. Congratulations, Ange.
Thank you.
Yay. Thanks, Ange.
Thank you.
That was great.
Nice. Oh, yes, this is awesome. I don't have pants on. Can I stop sharing my screen?